Hi, this is Alok, and today here I'm to talk about the production of DNA vaccines and some other aspects which are related with the DNA vaccines. So, when talking about the vaccine, the first question which shall come in our mind is that why vaccines are needed. We have plenty of medications available. Then, why vaccines? Can't we replace these vaccines by the some other medications, some formulations, like some simple medicines? So we shall understand that in developing countries, infectious disease generally cause 30 to 50% of all deaths. And SARS-CoV-2, which is known as the COVID-19, troubled has whole globe. As we know, there are many such diseases whose effective chemotherapeutic agents simply do not exist for. And this is why we have to go for vaccines. Talking about some successful stories of vaccines, so we shall understand two important vaccines importance like smallpox and diphtheria. So smallpox, it is an infectious disease caused by one or two virus variants known as variola major, variola minor. Both belong to the genus Orthopoxvirus. The last naturally occurring case was diagnosed in October 1977 and WHO certified global eradication of disease in 1980. So this was only possible with the help of vaccine against smallpox, which was developed by the in 18th century by the Dr. Edward Jenner. And it is said that 300 million people in 20th century and 500 million people in last 100 years were killed by this disease. Another disease where the role of vaccine is very important is diphtheria. Diphtheria is a respiratory infection which is caused by the Corynebacterium diphtheria, and this grows or white patches develop in throat of children which block the airway and leads the death of that child. This disease is most common in the sub-Saharan regions, and after 2015 it is said that this disease was ended or declined to very minimal case occurrence. And you know about DPT vaccines. Three vaccines are given together. All three vaccines are related to the respiration related infections like DPT, diphtheria, pertussis, and tuberculosis. So these two diseases were only curtailed when the use of vaccine was at very large scale. So let's talk about the problem with the traditional vaccines, because there are plenty of other types of vaccines like live attenuated vaccines, toxoid, and then subunit vaccine. Then why we go for DNA vaccines? So we shall understand that traditional vaccines are actually having some health related concerns, limitations, and this is why we go for the DNA vaccines. So here we will talk about some concerns which are with the traditional vaccines, our traditional immunization system. So one is the danger of reversion of virulent stage. So talking about the live attenuated vaccines or killed or weak strain attenuated vaccines, in those the weaker strain of a particular disease causing agent are inoculated in a human to induce the immunogenicity. But we are not sure that weaker strain can turn into virulent and cause many other concerns. Another concern is that live attenuated pathogen can produce severe disease in individuals with the immune system deficiencies. So these vaccines are not effective with the people who are having low immune defense systems. Suppose there is a person with the AIDS whose immune system is very low or very retarded, and if these vaccines are given, the weaker strain even can cause several problems in them. The principal component bacteria's outer membrane lipopolysaccharide, which is a lipopolysaccharide, also called as endotoxin, which may elicit a strong toxic response. So in many vaccines, we use the outer membrane protein of a bacteria to induce the immunogenicity, which in some cases can act as endotoxin and cause various other problems in the individual. And direct risk to workers who culture dangerous pathogens in large amounts for production of vaccines. This is another concern, and there is a possibility that organism or toxin may not be completely killed or inactivated in the process of making the vaccine, where the weaker strains are killed or attenuated, or some toxin which is inactivated. There may be chances that a particular thing is not permanently killed or inactivated. In that case, it may lead to several problems. Now talking about the DNA vaccine and recombinant vaccine. So DNA vaccine and recombinant vaccine follow various similar things, but they are two different entities, and let's understand how they are different from each other. So a comparison of the steps involved in the production of a DNA vaccine and a recombinant protein vaccine. Initial steps involve in generation of plasmid DNA encoding the protein gene of interest for both type of vaccines is similar. Here you can see the plasmid preparation or incorporating the gene of interest in both sort of vaccines is similar step or common step. For DNA vaccines, only expansion and purification of plasmid is required prior to immunization. But the production of recombinant vaccines requires additional steps for the expression and purification of protein itself. There we let that plasmid express, and that particular protein is used as a vaccine. But in a DNA vaccine, the DNA strand itself is used as a vaccine. Immune agent that can induce the immunogenicity is a DNA here. So these are some differences between the DNA vaccines and recombinant vaccines. Now we shall talk about the DNA vaccines, like how we design the plasmid constructs and other steps involved in this. So talking about the design of plasmid DNA constructs for vaccine, there are some criteria for this. So what are the criteria for a suitable plasmid? That is, the plasmid shall not be replicating in mammalian cell and shall not be integrating to mammalian genome. Another criteria is a DNA vaccine vectors shall have very minimal functions such that gene expression in E. coli is selectable marker and gene expression in mammalian cell is antigen. Only two sort of functions are must there. One is the expression in E. coli, and that is the selectable marker. Selectable markers would be expressed in the E. coli, and gene expression, like that particular antigen expression, shall happen in the mammalian cell. These are the two main functions of a plasmid construct that should be designed in this way. Additional plasmid functions such as LAC-Z and other genes should be removed because they have no role to play here. So while we talk about an ideal vector for DNA vaccines should be safe in humans and easily produced at commercial scale. That is the most important thing. The most important safety issue for a DNA vaccine vector is that the possibility of plasmid DNA integrating into human chromosome. To minimize this risk, what we do, the vector should not replicate in mammalian cells. Therefore, a vector should be chosen that does not contain mammalian origin of replication. The origin of replication should be such that it only replicate in a prokaryotic cell. means in the host cell like expression vector for selectable marker, or where we are going to produce the high number copies in E. coli. But there should not be origin of replication for the eukaryotic cells, like most specifically the mammalian cells. So to further minimize the possibility of vector integrating into human genome, the vector sequencing should be blasted. This plasmid or vector should be blasted into human genome by using some blasting sites, or there are various tools, and the human genome sequence to make sure that there are few if any strong homologs to the human genome. We shall sequence it and find out if there is any homologous or not. If there is a homologous, we have to slightly alter it, otherwise it will be integrating into the human genome and lead various genetic concerns. So large number of molecules per dose are required. Actually, in DNA vaccine, it is not that you produce 2 or 3 copies of DNAs and that you will incorporate in a human or in an individual, and that will be curing the disease. You should understand that we need like lots of amounts of that particular DNA vaccine, because when we are incorporating the DNA vaccine, it should be in such a level that uh, it at least enters or presents into like few thousands or few lakhs of cells, so that, and another concern is that key in a particular tissue, when this DNA vaccine is entering, the cells restriction system that will be degrading these, so there will be lots of DNA which will be degraded, and very few DNA strands of DNA vaccine will be there to get expressed as an antigen. So for this reason we need large quantity of DNA vaccine to incorporate in a human. Therefore, a commercially viable vaccine must give high yields of plasmid, preferably through a simple production process. For this reason, bacterial plasmids propagation is studied in gram-negative bacteria E. coli, because E. coli is the simplest host that can produce high number copies of these vectors. Talking about the PUC, this particular vector, this is a commercially available PUC vector or PVAX1 vectors, because these vectors are from the PUC categories. So PUC stands for the plasmid from the California University or University of California. And this yields between 500 to 700 copies per E. coli cell, and it is readily available. That is the advantage of this particular vector. And unlike col-even vector, there are other vectors like colicin even gene vectors. It provides immunity from expression of gene and also the mobility with a low copy number plasmid, but after degradation of some copies, it becomes high copy number plasmid and produce 1,000 copies and even more such vectors, and it is not suitable for a vaccine, because there should be replication, but not that high that there is a region there will be a possibility of that particular plasmid to integrate into the human genome. So there should be high number copies, around 500 to 600, but not that high like 1,000 or 1,200. In that case, it will lead some other concerns also. So this is a PUC vector or PVAX1 vector. This is a, in that category there are various vectors, and there you can see this so what are other things which should be in a plasmid DNA construct is that the mammalian promoter and poly-A terminated signal also need to be addressed. So in this particular, when this particular plasmid will be expressed in your mammalian cell, then there should be a have to have a mammalian promoter added in this plasmid, and a poly-A terminator, because in eukaryotic mRNA there will be poly A tail that should be terminated for for this particular mRNA to get expressed into a protein or antigen. The amount of plasmid that is internalized in vivo has been estimated to be in picograms. In vivo, even a picograms quantity is working, but in a cell we need to have more than this amount, like picograms, more than this. And it is found that the choice of transcription or poly A signal can have dramatic effect on the upon strength of a DNA. So the combination of CMV, the cytomegalovirus, and what promoter we add here? The cytomegalovirus promoter and bovine growth hormone (BGH) we add here. Bovine growth hormone. This is the poly A terminator. We add this in this particular plasmid. It provides a high level of transcription. If we add this, the promoter like mammalian promoter CMV, cytomegalovirus promoter, if we add, and BGS poly A terminator if we add, then this particular plasmid exhibit the high level of transcription. Now the vector contains PUC backbone. I told you, no, this vector plasmid were firstly made or constructed or developed by the University of California, and this is why this particular vector is known as PUC. And the CMV promoter with intron A. There should be a intron A, and the BGS terminator and kanamycin resistant. The kanamycin resistant, this is for what? This is for the expression or selectable marker in E. coli. How we will know that this particular vector is being copied in a particular host cells like E. coli. So that is why we add the kanamycin. That is the antibacterial element. So these are all the things which are very important for designing a plasmid DNA construct, and commercially available. I told, no, this PVAX 1 is a commercially available plasmid, and this only differ in not having the intron A. Here intron A is not added. That is only the difference is here. Otherwise it is quite similar, that PUC vector. Now we shall see the major steps involved in a vector preparation are generation of antigen insert. That is the first crucial step. So design primer with preparation of restriction map of antigen sequence and choose a transcription site in PVAX1 multiple cloning sites. This can be performed by use of restriction enzyme for isolation of antigen sequence and PFU polymerase enzyme in PCR to get plenty copies of antigen sequence cut purified DNA fragment by NHE1 and XHO1. These are the restriction enzymes which can cut the DNA fragment at a purification. So talking about the PFU DNA polymerase, which is known as the histidine, an enzyme found in the hyperthermophilic archaeon like Pyrococcus furiosus, where it functions to copy the organism DNA during the cell division. So when we are getting the multiple copies of this plasmid in PCR, then this PFU enzyme is used, and after that cutting, after we get the plenty of copies, we cut a purified DNA fragment by these two restriction enzymes. Ingestion of antigen gene into the DNA vaccine vector. So we cut the PVAX1 with the NHE and NHE1 and XHO1 and purify the vector by agarose gel electrophoresis. We cut these vectors by by their restriction side, and we separate this by the agarose gel electrophoresis. Recover the linearized vector using the QLA quick gel extraction kit. So there are various extraction kit are available, readily available. So we, as we cut this circular plasmid, so this becomes now linear, and now we separate it and set up ligation of vector to the antigen fragment, a molar ratio insert. A vector 6 to 10 into 1. So ligation can be done by the ROC rapid ligation kit. Those strands should be ligated, and in between also when we cut this plasmid we have to incorporate our gene of interest. That particular sequence can be added by this ROC rapid ligation kit and transform competent DH5 cells with the ligation reaction and plate onto L-kanamycin. We put these cells on L-kanamycin just for the marking, to getting know that whether this gene is getting expressed. If that particular gene is being expressed, then it will produce kanamycin, and we will know by the agar plates, like by the color change. I incubate these plates at 33 degrees C for 16 to 24 hours. Select well isolated colonies from the transformation plates and grow 5 ml kanamycin cultures. Make plasmid mini preps from the using QIA prep mini prep kit and screen the clones by restriction digestion by the NHE1 and XHO1. So where suppose there is a this selectable marker like kanamycin resistant gene is expressed, so those cells will be not dying even they are growing in a 5 microliter kanamycin cultures. In that culture, if it is these cells are cultivating, they will be not dying because they have the resistant genes. So this is how we will get to know that our gene of interest or the plasmid construct we made, that is being expressed. Now talking about the verifying clones by sequence and expression. We have to verify the clones. The clones, we high number of copies we got in E. coli. We have to check whether there is a exact sequence which we incorporated, or there is any mutation, or there is any false adding. So for that we have to check it. So how we will check? Clones that are identified restriction mapping. We shall have a restriction mapping should be further verified by sequencing the insert. This can be done by T7 promoter priming site and BGH reverse priming site present in the PVAX1 vector. Check for expression of sequence verified clones in transiently transfected mammalian cells line before using DNA vaccine in animal experiments. So before we go for the animal experiments, we have to check that the gene we incorporated, whether there is our gene of interest as such, or it is modified, or false heading, or any other mistakes are being occurred. A cell line such as rhabdomyosarcoma cells is good choice because it is easily maintained and transfected. So this can be done only, we have to, how we will check this? We will be the transfecting these RD cells, and then we will be sequencing like T7 promoter priming site and BGH. With these two priming sites we will sequence them, and we will get to know that whether the gene is as such or not. Selection of E. coli strain for plasmid production. Many E. coli strains are commercially available as competent cells and have been developed for specific needs in molecular biology. Most strains have been characterized for protein expression, not for plasmid stability and production. That is the biggest concern here, because all the E. coli strains are available today. Mostly they are protein expressions hosts, but they barely are manufactured for plasmid stability and production. Therefore, several strains should be tested prior to choosing a host strain. Once several candidate strains are selected, each should be tested with the final vaccine clone to determine which will be grow at reasonable rate 2 or less doubling time, achieve a high cell density and give the highest plasmid DNA yields in the fermentation medium in large scale production. So now we shall talk about the antigen expression system for plasmid DNA vaccines. Often foreign proteins, especially small ones, occur in minute quantities when they are produced heterologous. So when we are producing the particular protein in non-specific host, then there will be quantity decline, and that is what happened in case of DNA vaccines, because when the plasmid is incorporated in the mammalian cell, that is non-specific host cells, and that is why there is a production of this particular antigen proteins is very minimal, and that is why the apparently low level of expression is in many instances actually due to the degradation of foreign proteins, because these proteins are not the proteins of that particular host cells. So the enzymes, particularly various kind of protease enzymes, will be degrading these foreign proteins, and this is biggest drawback in the DNA proteins, DNA vaccines. One way to solve this problem is to engineer a DNA construct that encodes a target protein that is in frame with in frame with a stable host protein. We can make such protein or target protein along with that your antigen protein that is stable with the host protein. We have to, for this purpose, we have to engineer our DNA plasmid construct. This combined single protein, which is called as a fusion protein, this will be known as a fusion protein because it will be having a protein that is a stable host protein and the antigen protein. These proteins will be fused together. This fusion protein protects the cloned gene product from the attack by host specific proteases. Another way is to genetically engineer the best competent E. coli cells so that overexpression of rare tRNA happens. In this way, the high possibility of expressing the antigen gene, because we know that cellular incompatibility that can interfere with the efficient translation occurs when a cloned gene has codon that are rarely used by cells. Another way can be that we have to genetically engineer competent E. coli cells, and which will be having the rare tRNA cells which are not utilized by the host cells normally. But when this plasmid DNA construct is incorporated, that tRNA will be used by that particular plasmid, and this is how the overexpression we can expect. So this is the overall view. This is a way like this particular gene of interest is incorporated and in a plasmid, and this is a plasmid construction, how we will construct a plasmid. And this is for the expression like, okay, so E. coli host cells like plasmid with foreign gene and low level of foreign gene production, because you know that there will be degradation will be happening. So what we can do is there are two ways, and one way is represented here, that we have to put some overproduced tRNA. These tRNA are very rare kind of tRNA, and that will be not utilized by these E. coli cells, and this plasmid with foreign gene will utilize this tRNA, and high level of foreign gene expression will be occurring. Now we shall talk about the successful DNA vaccines till date. Already we have talked about the vaccines importance, but those were not the DNA vaccine. So now we have to talk about the successful DNA vaccine up to date. So in August 2021, Indian authorities gave emergency approval to Zycovid-D, developed by Candela Healthcare, is the first DNA vaccine approved in humans. DNA vaccines against cancer are under development which can become potential immunotherapy against various kinds of cancers. DNA vaccines expression of IL-2. So IL-2, which is a interleukin 2 and heat shock protein 65. So interleukin 2, this is a type of cytokine signaling molecule in the immune systems, and HSP 65, heat shock proteins which protect this antigen proteins. So fusion genes was studied. So we studied the expression of IL-2 and HSP65 fusion gene, and it elevated the immunogenicity and protective as well as therapeutic effect of HSP65 DNA vaccine against tuberculosis in mice. And HIV negative people were used to study the effects of preventive vaccine candidates to see if they can prevent infection. Many scholars see DNA vaccines as future possible immunotherapy against HIV. DNA vaccine is one of the novel approaches for developing new generation vaccines against malaria. Coated DNA vaccines have been shown to exhibit good immunogenicity and show protective levels of antigen-specific immunoglobulin G and elevated proportions of CD4, CD8 T cells, interferon gamma, interleukin 12 levels in the serum, and cultured splenocyte, supernatant, as well as interferon gamma producing cells in spleen. So our current understanding is that DNA vaccine induce innate and adaptive immune responses in two ways. These are the two ways. The first one is that encode protein antigen by DNA plasmid antigen, DNA plasmid. This is the DNA plasmid encoding protein region, gene region, which can be expressed in the stromal cells, these muscle cells, and as well as in the DCs, the dendrocytic cells, where these antigens are processed and presented to naive CD4 and CD8 cells, either by direct or cross penetration. So when this particular DNA vaccine will be transfected in a muscle cells, it will lead to the expression of antigen, and these antigens will be targeting to either by the direct penetration or by cross penetration to these called to the CD4 cells and CD8 cells, and they will produce immunoglobulin G and other interferons or other cytokines or to induce the immunogenicity. Another way is that the transfected DNA plasmid itself may bind to unidentified cytosolic DNA sensors and activate the TBK1 and STING pathway, and production of type 1 interferon, which functions as adjuvants. It will produce the transferons which will be acting against that antigen, and this is how the immunogenicity will be produced in a cell. So now we have talked about that how the DNA vaccine can be produced, but the more bigger challenge is that how these DNA vaccine will be delivered to a human tissue. So needle-free injection of DNA vaccines and surface modified biodegradable microsphere of DNA vaccines delivery, and a dendrimer like DNA based vector for DNA delivery. These are some methods by which the DNA vaccine can be delivered. So needle-free injection of DNA vaccines. Needle-free injection is accomplished by forcing liquid medications at high speed through a micro orifice. This will be a instrument which will be having the micro orifice to the similar pore size as our skin have the pore size. And Beclard and Galante of France has developed the first needle-free acupuncture device which delivered liquid medications at pressure of 25 to 30 atmospheric without the need of a needle. Needle-free jet injectors are approved for human use. Several different jet injectors have been used to deliver DNA vaccines. Mucosal immunoglobulin V was induced by HIV-1 DNA vaccines when mice were immunized by intraorally using a jet injector. The jet injector name is CyriJet Mark II, designed for the application of dental anesthetics. Bioinjector utilizes the compressed CO2 to force the liquid through a micro orifice, and it can deliver vaccine to traditionally targeted tissues such as skin, muscle, adipose without the use of needle. Another method is surface modified biodegradable microsphere for the DNA vaccine delivery. Encapsulating DNA within degradable delivery vehicles such as micro or nanosphere provides an effective way to protect the DNA from the surrounding environment prior to the delivery. One means of highly specific cell targeting is through the addition of vehicle surface of ligands that binds specifically to the receptors on the surface of target cell types. This is one way. And chemical conjugation of ligands to the microsphere surface requires a selection of appropriate conjugation chemistry. When using microspheres that are biodegradable, an ideal conjugation scheme minimizes premature degradation of the microspheres. For this method, major requirements are PLGL, which stands for the lactic or polylactic co-glycolic acid, and which have broad application in target drug delivery. These are stored at minus 20 degrees C, protected from moisture. And dichloromethane is another such material. DNA to be encapsulated. Another method is a dendrimer-like DNA-based vector for DNA delivery. A synthetic polymer that is anisotropic is needed. The recently developed dendrimer-like DNA is a true dendritic polymer that can be anisotropic, thus providing a great potential as novel vector to carry multifunctional modules that target specific DNA obstacles. So here is that we are not using plasmid, but we are using the some what called this J sort of DNA structures, and in those structures the gene of interest is incorporated, and that will be incorporated. In that case, there will be a way that directly they will be attaching to that particular DNA and bringing those DNA to the host cell or mammalian target cells. DLDNA is capable of carrying many various peptides, especially those peptides derived from the viruses. These viral peptides can perform desired biological function, including cellular targeting, DNA condensing, endosome disrupting, and nuclear targeting. Pre-made modules will further increase the flexibility and make plug and play. So there is a plug and play mechanism possible. Such flexibility is particularly useful in studying the complex process of DNA delivery because little is known quantitatively about the intracellular events, and one can easily adjust the delivery vector based on the experimental outcomes. So here we can make this possible, and in addition the DNA system is capable of carrying both genes and antigens as well as other entities such as enzymes and chemical drugs. So these are all three methods. This is the biojector, this is the microsphere which is made of the PLGA. So it will be having some sites where the DNA vaccines will be fitted, and these DNA vaccine will be protected from the outer environment. And last one is the dendrimer, like DNA vaccine delivery. So dendrimer with the target gene, it will be having target gene will be within that, and attached to the cell surface, and by the endocytosis it enters within a cell, and there it releases the its DNA to the nucleus, and there it mRNA produced, and mRNA is released to the cytoplasm, and there it get expressed. The common phenomena, as you know, that transcription and translation. So as we know that DNA vaccines, though they seem very promising, but still there are limitations. The one limitation is that they are not that very effective in mammals. These DNA vaccines have been working efficiently in the research when they did a they did a trial on the mice and other mammals. But in humans, these DNA vaccines are not that effective. And to enhance the activity of these DNA vaccines, we need to add some other adjuvants. So let's see what are these adjuvants. The activity of bacterial DNA, or the synthetic DNA, oligodeoxynucleotide, which is known as CpG-ODN, that mimic it is the first characterized in mimic. These are the CpG-ODN molecules, one of the enhancement agents for the DNA vaccines. So, they stimulate a polyclonal B cell activation characterized by cell proliferation, reduced apoptosis, secretion of interleukin IL-6, IL-10, and polyclonal immunoglobulin M, and upregulation of co-stimulatory molecules such as class 2 major histocompatibility complex MHSC, CD80, CD86, CD40. So these are the molecules which enhance the DNA activity, and we shall understand that this CpG-ODN also act as a professional antigen presenting cells like APC, monocytes, macrophages, and DCs, increasing their antigen uptake and presentation, as well as augmenting the expressions of co-stimulatory molecules and the secretion of pro-inflammatory cytokines in turns activate NK cells, CD4, CD8 plus, mu ST cells, cytokinins, and they enhance the lytic activity and secretion of high levels of interferons. So these CpG-ODNs are very important in the cell DNA vaccine enhancement. So we shall understand what are these CpG. So CpG are the CD sites, are regions of DNA where the cytosine nucleotide is followed by guanine nucleotide in the linear sequence of the bases along its 5 to 3 prime direction. CpG sites occur which have high frequency in genomic regions called as CpG islands. These CpG can be methylated to form 5-methyl cytosine, and the enzyme which add this methyl group are called as DNA methyltransferase. In mammals, 70 to 80% CpG cytosines are methylated, And this is the one benefit that we can add these CPG or dioligonucleotide in a specific sequence so that the DNA vaccine which is delivered to a human tissue is not being degraded. And this is how we can enhance the activity of these sort of proteins. This is the overall procedure how it can happen. This is already I talked. And these are some of my references like DNA vaccine methods and protocols, second edition by Human Express, DNA Pharmaceuticals by Martin Scliff, current topics in microbiology and immunology, molecular biotechnology by Bernard or Glick et al. By this, I'm ending my presentation. Thank you so much.